Let us now consider the microscopic features of muscle tissue. I am grateful to Professor Michael Hodge of University of Michigan Medical School for providing the images of histology sections used in this video. Muscle tissue is made up of aggregates of specialized elongated cells that are capable of contraction to bring about movement or change in the size or shape of internal organs. Muscle can be classified as striated muscle, which has regular arrangement of myofilaments, and non-striated muscle, which has irregular arrangement of myofilaments. Striated muscle is further classified as skeletal muscle and cardiac muscle. Non-striated muscle includes smooth muscle. While skeletal muscle is considered to be under voluntary control, the cardiac and smooth muscle are considered to be involuntary. Skeletal muscle is made up of long muscle fibers held together by collagenous sheaths. These fibers are multinucleate because they are structural syncytium developed by fusion of about 50 to 100 myoblasts. Skeletal muscle fibers appear like long cylindrical structures with prominent transverse striations. Their cytoplasm, known as sarcoplasm, is filled with regularly arranged myofibrils, and it is surrounded by sarcolemma or the plasmalemma. These fibers are multinucleate, showing peripheral, moderately euchromatic oval nuclei. This is a magnified view of skeletal muscle, showing striations and peripheral nuclei. This is an electron microscopic view of skeletal muscle with a magnification of about 32,000 times. We are seeing pale eye zone, which is bisected by the Z line, which represents the site of anchoring of actin filaments. These eye bands are alternating with dark band, or otherwise called as A bands. Within the A band, there is a central pale H zone, which is the area where the actin filaments do not intervene between myosin filaments. And this H zone shows a central darker M line, which represents the site of anchoring of myosin filaments. The distance between two Z lines is called as a sarcomere, which is the functional contractile unit of skeletal muscle, measuring about 2.2 microns. Muscle also shows muscle triad, which has a central transverse tubule, which is the tubular invagination of sarcolemma, and this is flanked on either side by terminal cistern. The wave of depolarization which travels along this transverse tubule will result in releasing of calcium ions from the terminal cistern, and that will result in contraction of the muscle fiber. When we see the cross-section of skeletal muscle, we are seeing uniformly sized polygonal muscle fibers with stippled cytoplasm because of myofilaments and peripheral nuclei. Individual muscle fiber is covered by endomysium, which has capillaries and nerve fibers. Bundles of muscle fibers or fascicles are covered by perimysium, with larger blood vessels, nerves, and neuromuscular spindles. Epimysium covers the entire muscle, and it extends between groups of fascicles. Now let's look into the features of cardiac muscle. In cardiac muscle, the muscle fibers are arranged in various directions. Endomysium is less regularly arranged around individual muscle fiber. We can see coarser perimysium between the fiber bundles. Individual cardiac muscle fiber shows 1 to 2 central nuclei surrounded by sarcoplasm, which has got abundant cellular organelle. Cross striations are seen due to regular arrangement of myofilaments. These fibers are branching and anastomosing, and we see the presence of intercalated discs. Intercalated discs represent junction between two adjacent cardiomyocytes. When we see it under electron microscope, there are transverse and lateral portions in the intercalated discs. The lateral portion has gap junctions in them, which will allow electrical coupling so that the cardiac muscle functions as a functional syncytium. Cross striations in cardiac muscle are not as prominent as what we see in skeletal muscle. That's because these contractile proteins are within the mitochondria-rich sarcoplasm, and myofilaments are comparatively less well delineated. Here we are seeing cardiac muscle under electron microscope. We can see the electron intercalated disc. On the left side, we are seeing the stepladder pattern of intercalated disc arrangement. We are seeing both transverse and lateral portions to it. In addition, we are seeing the A band with H zone and I band with Z lines. We are also seeing lipid droplets in the muscle fiber. Smooth muscle has no regular arrangement of myofibrils into sarcomeres. Therefore, it does not show striations. It's found in the wall of viscera as well as in the wall of blood vessels. Smooth muscle fibers are smaller spindle-shaped fibers with tapering ends. They have a central single cigar-shaped nucleus. In the perinuclear cytoplasm or sarcoplasm, we find abundant organelle, and myofilaments are usually missing in that region. In cross-section, we see circular fibers of varying size depending upon the level of section. Nucleus is present only if the section passes through the center of the fiber, and each cell is covered by basal lamina. This sarcolemma shows cup-shaped surface invaginations known as caveolae. Because of the presence of caveolae on the surface, the smooth muscle fibers do not show T-tubules or triads on their surface. So, quickly recalling what we have seen so far, skeletal muscle fiber is a structural syncytium with multiple peripheral nuclei and prominent cross striations. Cardiac muscle is a functional syncytium maintaining ionic continuity across the gap junctions at intercalated discs. It has a single central nucleus and less prominent cross striations. Smooth muscle fibers are non-striated, spindle-shaped fibers with a single central cigar-shaped nucleus. Thank you. Hope you enjoyed this video. You can also visit this site for similar histology videos.